Hi, I am one of the ULAs for B-Side 222, and this is an Exam 1 review covering the topics of chromosomes, mitosis, and meiosis. So, let's get started. So, a genome is a set of instructions that an organism possesses, and it is organized into DNA molecules. These DNA molecules are then further organized into chromosomes. And we've seen chromosomes pictured like this. And it is made up of the centromere in the middle, the telomeres at the end, and consists of two arms. So you have the P arm, or the short arm, and the Q arm, or the long arm. And in class, we've discussed ways to identify chromosomes, which include shape, size, and pattern. And there's 4 shapes in particular that we've discussed. So you have the metacentric, where the centromere is located in the center, and you have the P and the Q arm about the same size. The sub-metacentric, where the centromere is located upper, middle, and resulting in a shorter P arm and a longer Q arm. Acrocentric, where the centromere is located very close to the top, and you have very short P arm. And finally, telocentric, where the centromere is essentially located at the very top of the chromosome. And I've received a few questions on exactly what is a chromosome or a chromatid. So just for clarification, this is considered a chromosome, and this is considered a chromosome. This chromosome consists of 2 sister chromatids. And these sister chromatids possess the same genetic information. So once a cell undergoes DNA replication, it's presented like this. So these 2 chromatids are held together by cohesin, and once replication has occurred, mitosis has occurred, then they will result in this. And this is still called one chromosome, as well as this one, except with the exception that this one consists of 2 sister chromatids. I hope that helps clarify any confusion. So this leads us into mitosis. So prior to mitosis, a cell would be an interphase, and interphase consists of several stages. So at the beginning, you have the G1, which is cell growth, and then it will go into G1S checkpoint, and it will essentially ask the question, am I ready to replicate? And if it gets the green light, then it will go into synthesis, and during synthesis, this is where DNA replication occurs. So once DNA replication has occurred, you have G2, where the cell grows even more and produces proteins to prepare for later use, and it goes through the G2M checkpoint. And during this checkpoint, it is essentially asking, are all the DNA undamaged and intact? And if it gets a green light there, then it goes into mitosis. And with many cancers, these checkpoints are not necessarily obeyed, which is why it would result in uncontrolled cell growth. So we have prophase, which occurs first, and we can see the chromosomes condensed and pictured like this. And after prophase, we have the prometaphase, and during this, so you would have the centromeres. At the centromeres are kinetochores, and the microtubules will attach to the centromeres, and then that would lead us to metaphase, when the chromosomes would line up across the middle. So you have the microtubules attached to the kinetochores on the centromere. And then metaphase is followed by anaphase, and that is when the microtubules move the chromosomes to their respective sides. And then that is followed by telophase, and then cytokinesis, where the cells are where you would result in two identical sister cells. And there are differences there are huge differences between mitosis and meiosis. So during meiosis, you undergo crossing over in prophase 1. This does not occur in mitosis. So the crossing over increases genetic diversity. And then it would go through the respective phases, so metaphase, anaphase, telophase 1, and then it would undergo a second process, but there is no synthesis that occurs again, which allows at the very end of meiosis to result in 4 haploid cells. So mitosis, you end up with 2 identical diploid cells, but in meiosis, you would end up with 4 haploid cells that are different from each other, amongst many reasons, including crossing over, which increases genetic diversity. I hope this video was helpful, and if you have any more questions, please refer to the lecture slide, the PowerPoints, or come up and ask the ULAs or the professors any questions. For understanding meiosis and mitosis and their differences, there's image that Dr. P had on ELMS, which is very helpful because she draws it out. And I said if you still have any confusion about that, refer to it. Thank you for watching.